Hello everyone, welcome back to my channel. Today we have a somewhat educational video on cell death. This is one of my main interests and what got me into going to graduate school in the first place. And my main interest is of course anything related to cell biology and molecular. And this was what kind of piqued my interest in going into cancer research, primarily because I believe the pathways themselves are really interesting, and the fact that going over some random ass material, there are a lot of ways cells can just die, not just eukaryotic cells but also bacterial cells as well. So all this is really important if you ever want to get a degree in microbiology and want to go into courses dealing with elementary microbiology, immunology, or even bacteriology. This is probably a good place to probably pique your interest. So what I'm going to discuss is apoptosis, which is a form of programmed cell death. So what apoptosis is it's a signal, it signals death of a cell from either inside or outside. The reason why we have apoptosis is because cells die constantly every single day. I'm talking in the millions, maybe in the billions. They are really useful in development, in case like whenever they develop, say like webbings on our feet, all those webbings kind of just get destroyed during the process of development. Also in our hands as well. It also is initiated if the cell is not needed. If the cell, say, is old enough and they're trying to make room for new cells that might be working more properly, those will be destroyed. If the cell is not functioning properly, whether it's because it's damaged or it starts attacking the body in some way, those will be programmed to be destroyed. Cells can be infected by a virus, which can cause a lot of damage to the other cells in the body, or it's also used to prevent cancer. There's a balance between life and death. In this case, is whether cells should continue dividing and proliferating, or there might be too many cells dying, which both extremes are really bad in our body, and is trying to maintain this balance. And another thing is to know is that any cancer cells have developed or evolved in a way where they have evaded apoptosis in some manner, way, or form. That's why there's still a lot of research being done in this field, trying to find out ways to inhibit these mechanisms, or understand these mechanisms a lot more in a lot of cancer cell lines that we have to test. And so in apoptosis we have two main pathways to go about. We got the intrinsic and the extrinsic pathway. The intrinsic pathway is what we'll be mainly going over today in today's video, and this is just apoptosis or programmed cell death that happens inside the cell. The extrinsic pathway, which will be the next video, will discuss what the cell death, apoptosis, that is a signal that's coming from outside the cells, which is usually by some sort of immune system, particularly a macrophage. And then just to clarify, I got kind of like two locality words, just because I'll be saying these a lot: pro-apoptotic proteins and anti-apoptotic proteins. Just by the names, pro-apoptotic proteins, these are particular proteins or enzymes that would promote apoptosis or promote cell death. These are pretty much the main things that initiate the entire pathways. While the anti-apoptotic proteins, these are, of course, they're still useful, but in some ways can be detrimental to the body or to the cell, depending on how it's upregulated or downregulated. Pretty much they inhibit cell death, and which allows cells to continue to grow and divide. So yeah, let's just, with this general synopsis, let's go into the intrinsic pathway. So I guess I'll be using my trusted pen for this. So what we're going to be discussing right now is the intrinsic apoptotic pathway. So this is the cell will be undergoing apoptosis. The signal in this case will be inside the cell, rather outside. The next video I will be discussing the extrinsic apoptotic pathway, since that one's a little bit more complicated to this one, but I feel like I need to at least explain this and then go to the extrinsic, because they kind of coincide with one another. But anyways, so as we know, apoptosis, programmed cell death, PCD, We have some we call them death signals or death stimulus that will cause the cell to undergo programmed cell death. It can be, in this case, double-stranded DNA break, hypoxia, and any type of intracellular stress, damage, and oxidative stress and damages. Whenever this is actually indicated inside the cell, there'll be a lot of these particular intracellular molecules that'll be used as signaling mechanisms to initiate apoptosis. First things first, whenever you see this type of shit going on, you have this serine threonine kinase, which will actually activate one of our main tumor suppressor proteins or genes. In this case, it's p53. This will essentially phosphorylate to the tumor suppressor and make activated to initiate all the pro-apoptotic proteins that you're about to witness right now. And another thing to say is that p53 tumor suppressor, like this name is supposed to say, it's meant to suppress cell proliferation. The thing about this particular gene, it is an oncogene, which it has the capability to cause a cancer. Depending on how it's mutated, it might be deactivated when it's supposed to be activated, or it might allow the cell to continue going through the cell cycle continuously without even stopping. And particularly the ATM serine threonine kinase is also used in regulating the cell cycle, not just cell death as well. It's possible that a cell it's trying to keep it into like the G1, G0 arrest. Once it's initiated, it will actually finally go through this entire process, because a lot of the tumor cells usually undergo such a thing. So once we got the p53 tumor suppressor, this will initiate these particular modulators of apoptosis. We got PUMA and NOXA. We know PUMA is the p53 upregulated modulator of apoptosis. These proteins has kind of like two functions. One, it actually activates or upregulates these BCL2 family proteins. They're called BAC, and there's even other kinds, but these are the primary main ones. And also inhibits this BCL2 anti-apoptotic proteins. They also come in extra large if you wanted. And it also comes in different forms and varieties. But the main point is that this has pretty much a function to upregulate these pro-apoptotic proteins and downregulate these. So what happens is, the reason why this is the pathway, this intrinsic pathway is happening inside, is the main target is just primarily because of the mitochondrial outer membrane. These the BACs and BAC will actually go and try to attach themselves into these little things called the MAC, which is just essentially a pore for them to open up the mitochondria, and this comes out as the mitochondrial outer membrane permeabilization, or the MUMPs. Creates this pore in the outer membrane, so it's essentially a hole in it. The hole is I just call it a MUMP pore, but some other people call it a voltage dependent ion pore. It's a little bit fancy, or BDAP. Yeah. So from here, what the main point is, once it's opened up, what happens is, see this cyto C? This is actually a very, very important electron carrier in electron transport chain. If you went into fucking general biology, this is used whenever you look at to transfer electrons from complex 3 and complex 4 in the electron transport channel in the fucking outer membrane of the mitochondria. So with the creation of this pore, it allows the cytochrome C to be released from the mitochondria, and you'll see what happens after this happens. Another thing that's also released, which I was going over, there's also the second messenger of apoptosis with SMAC and a Diablo homolog, which has extra functions of actually inhibiting BCL2, an anti-apoptotic protein. And there's also other proteins that you will also see in the extrinsic pathway which also benefits this. The again, the role of BCL2 is primarily to kind of form a dimer with these two, and by doing so, it inhibits the function of BACs or BAC to actually undergo apoptosis in this particular pathway. So the more cancer cells usually will have this upregulated much more compared to any other like somatic cells that you see inside a human vessel. Okay, so this is where we left off. We left off where the cytochrome C was released inside the cytosol of the cell. So what happens from there once it's released? Well, it starts recruiting these other proteins called APAF1. This stands for Apoptotic Protease Activating Factor 1. It kind of forms into this oligomer. Back when I was doing research about this, I read it as a heptamer. So there's usually like 7 subunits surrounding it. These little lines right here. But I'm assuming it comes in different forms as well. But this happens once these form with, as well as D80ADP, it forms into this structure known as the apoptosome. This apoptosome structure will actually begin the entire process of apoptosis by doing something known as the Caspase Cascade. The caspases are these protease enzymes which are made entirely of cysteine and Episodic amino acid residues primarily, and what they're used for, they usually come into these procaspase stage before being activated to initiate this cascade. So what happens is, with the help of the apoptosome, you got the procaspase 9, which is I guess you can say its deactivated form. Once it gets activated, the caspase will begun. Normally, whenever I went over this in like videos and such, I usually see these caspase enzymes kind of like I guess like if you've seen Beyblade, you got the top and then you got the little spinny thing right there. Once you spin it, it activates, so it kind of starts rotating around. This becomes procaspase to caspase 9. This is the initiator caspase of this pathway. From there, it will then, the reason why it's called a cascade, it will activate another caspase. In this case, we have the procaspase 3. This will be activated and become caspase 3. This caspase is what causes the cell death. This is known as the executioner caspase. From there, we will start recruiting pretty much DNases, which will cut the DNA into many small fragments until it's like you can't really see anything left of the cell, and as well as proteases. This is also proteases as well. It will begin to recruiting some cytosolic and nuclear proteins. Proteases, I don't know why the fuck I put proteins, but just know that these will be cleaved and will be destroyed inside the cell, as well as the DNA. From there, the cell will eventually die. This is a little short drawing I did. Imagine this is a cell, so any type of eukaryotic cell. During the time it's undergoing apoptosis, it will start blobbing up with these little forms around it. This is just, of course, it's just called blobbing. After a while, it will actually just dissipate into these little ball-shaped bodies. These are actually called apoptotic bodies. Once the cell has initiated and finalizes its programmed cell death. During this time, this is supposedly a non-inflammatory way of dying than necrosis. Necrosis, of course, is something that's more inflammatory. The reason why this is non-inflammatory, these is a really clean way of killing the cell, because once you have these apoptotic bodies just around the extracellular region, these will get pretty much phagocytosis by any type of immune system. Primarily the macrophage will go ahead and consume it. So it's a very clean process overall. Another thing I would like to mention is these caspases, particularly caspase 3, the main executioner caspase, can be inhibited by IAPs. IAP stands for inhibitors of apoptosis proteins. They come in, I know one of them is the XIAP, IAP. BCL can also count as an IAP protein. It's just a different family. So a lot of these cancers usually will have these also upregulated, so that way it can even though it goes through the entire process of getting the cytochrome C out of the mitochondria of the cell, it can do this cascade. This is the main thing that's required for apoptosis to even occur. So once this is inhibited, the cell just won't die at all. You may see signs or some assays where if you want to determine the membrane potential of the cell, if it's lower, it's always saying, well, it's the electron transport chain isn't working because the cytochrome C is out of the mitochondria. That's not supposed to be. However, the cell would still be living because it hasn't initiated this cascade at all. With that being said, there are some pathways that's also that can pretty much is caspase independent, doesn't require it. However, it is kind of a inflammatory response. We will see that with the next programmed cell death, which is known as Necro Apoptosis, which involves using some different type of kinases, appropriately called RIP kinases, and other particular enzymes in this. And again, we don't use these caspases since most of the time they will be inhibited by some means in most cancer cells. So this is just a quick overview of the intrinsic apoptotic pathway. In the next video, we will discuss more on the extrinsic and how they kind of relate with one another. Thank you all for watching. You all have a great day. Pew!